Hello, my name is Joris Veltman, and I will be introducing you to personal genomics. I will discuss the human genome and how we can read the human genome, talk to you a little bit about how we can interpret this information for personal genomics as an introduction for other speakers to follow up on. I'm a director at the Institute of Genetic Medicine in Newcastle, and I also have a joint appointment at Viome Inc in Nijmegen. When you talk about personal genomics, of course, you first have to look at the genome, which is an enormous amount of DNA letters, 6 billion in each of our cells. Importantly, most of the genome of two individuals is very similar to each other, but there are also lots of variations, and these can be individual nucleotides changing, but there could also be large deletions in the DNAs, inversions, repeat expansions, etc. So in order to really understand the personal genome, you have to identify all of this variation in order to start trying to link it to disease, for example. So if you would compare the genome of two individuals, and you see two of them here, you actually will see that they are mostly similar. The genomes are very similar at most positions, but still there are about 4 million positions in the genome which are different. And those positions are interesting when it comes to personal genomics. When you think about personal genomics, I think there's two major challenges. One is the identification of all of the genetic variation. As I already mentioned, the genome is very big, so it needs to be done very fast. You have to identify this variation very quickly. It has to be done affordable if we want to do this widespread in the community. And it has to be done very accurately. And because I already mentioned there's many different types of variation, it's also important to realize that some variation is more difficult to detect than others at this moment. So we still need improvements in the technology in order for the variation to be detected. At this moment, not a single genome has been sequenced perfectly. There's still, in every genome that has been sequenced, lots of variations missing. Once you have that variation, it's all about interpretation. And interpretation is not done by comparing groups of individuals and saying, well, there's differences between these two groups. Now for personal genomics, of course, you have to do it in individuals. You have to do it in individual cases who need to have reliable and useful information. And it's important, again, to realize that it depends very much what you want to do. Do you want to diagnose the genetic cause of a rare Mendelian disease? And actually, genome sequencing is already very useful and is being actually applied in the clinic very much. When it comes to predicting the onset of a complex disorder where genetics plays perhaps a rather small role, it's much more difficult. So a lot has changed in genetics in the last 20 years. This is when I started my PhD in 1989, and you see that genetics was really an art. You had to be trained in recognizing chromosomes. Now in 2018, genetics is an industry, and here you see, and this is already an outdated picture, because of the sequencing machines that you see, but actually you see the machines are large-scale sequencing of genomes. The genetics laboratory is much less of an art. It's just really preparing samples to be put into these kind of big machines. What has happened is a very big change in the way we read our DNA, and therefore this has been called next-generation sequencing. And it's very different from traditional sequencing in that with traditional sequencing, individual fragments were sequenced. But because of miniaturization and parallelization, next-generation sequencing allows us to accurately measure millions of DNA fragments simultaneously. And this has really speeded up the whole process of DNA sequencing, but it has also made it much more affordable. We can do it in very small volumes. And what you see here is one of the costs dropping of sequencing an entire human genome from hundreds of millions a little over, let's say, 15 years ago to now less than about $1,000, depending on, let's say, the quality of the genome that you are looking at and the speed that you want the data to get to you. But this means that personal genomes are now becoming affordable, and we can really use these in the clinic, and we can use them for personal genomics. So these are the kind of ways to visualize a genome. On the left, you see the genome of a patient with intellectual disability, where there may be 1 or 2 mutations in the DNA that cause the disease. Whereas in cancer, because of genetic instability, you see that there is a lot happening, and it's really fascinating that these cells are still able to divide. Now, because we have now the ability to read genomes in the clinic and in personal life, there's lots of things you can do with it. And here I've just done some pictures of moments in your life where genomes could be useful, either, for example, during the screening of parents-to-be, until, let's say, all the way back to when you have cancer and you want to have a personal treatment for your cancer. We'll give you one short example of our own work where we used next-generation sequencing to identify the cause of a rare genetic disease. In this case, this was a rare sporadic disorder. So a patient was born in healthy parents, and it was very difficult to identify the cause with traditional technologies. But what we did in this case, we sequenced all of the genes in this patient with this syndrome, identified about 12,000 variants in genes, of which about half of them affect proteins, and we saw that about 180 of those are not varying in the normal population. So we thought those would probably be the ones that can cause a severe disorder like Schinzel-Gideon syndrome. Because we had sequenced other patients with the same syndrome, and we expected that patients with this syndrome would all have a mutation in the same gene, we could actually filter for genes with variation in all patients with the same syndrome. And then actually by doing this in 4 patients, we could immediately identify the gene that was mutated in this case. So without knowing anything about the genetic cause of this disease, in one experiment using next-generation sequencing in 4 patients with the same syndrome, we could actually identify the genetic cause of the disease. Now, of course, you have to further follow this up. One of the important things is that the parents of these children do not carry the mutation, so the mutation occurs de novo in the germline of these patients, and that, of course, is an additional proof that this is the cause of the disease. These kind of applications have been initially done in research, but now are being used widespread throughout, let's say, the diagnostic arena. And I've recently moved to England, and in England now 100,000 genomes are being sequenced as part of routine clinical care. So it really shows you that genome sequencing is becoming integrated into routine care, mostly for rare genetic disease as well as for cancer. Now, interestingly, when it comes to personal genomics, is that you can use this also for many more common traits. For example, for bipolar disorder, for diabetes, for rheumatoid arthritis. So diseases where many people in the population suffer from, and diseases that some of us may develop later in life. And of course, this is important if you want to predict by looking at your genome whether you are at increased risk or not. Important here is to realize that the effect size of many of the variants identified in genetic studies is often modest. So you really have to combine multiple variants in order to see a difference between persons without a disorder and persons who get a disorder. Interestingly, it's not only about severe diseases, but also other common traits, behavioral traits, extraversion, neuroticism, openness. Also, these kind of traits have been associated with variation in the DNA. Now, the genetic confidence in which these are linked is much less strong. So it's important, of course, that this is scientifically replicated, and this is one of the studies where so far it's just showing you that yes, there is data there, but it's not replicated. So you have to be very careful in the interpretation of these kind of variants. But by expanding this to larger cohorts of patients, there are maybe genetic variants that really do something and can be used for predicting how a person is. But again, you have to be careful in the interpretation. So finally, I've looked at my own genome in 2010. What you can see when you look at the profile from 23andMe, one of the direct-to-consumer companies that I did, is that you see that actually I have a variant in the DNA that would require me to use a lower dose of a blood thinner, warfarin, but also they say that based on some of my DNA variations, I should avoid raw oysters. And have to be honest, I love oysters and eat them all the time. In addition, the profile says that I am at a higher risk for a condition called celiac disease. Interestingly, if you look what that higher risk means, it actually means that instead of having a risk of 0.1 out of 100, I'm at a risk of 0.3 out of 100. So it means it's a very small risk. And it also says that I'm not a carrier of any of the inherited disease variants tested by 23andMe, but actually there's very few tested. There's many more. So take-home messages, technological innovations allow for the reliable and affordable analysis of individual genomes. So really technology is allowing us to start looking at genomes routinely in the population. It's used in patient care, mostly for rare disease and for cancer. And the opportunities of genomics to start predicting and preventing common disease, or for example, personalized drug treatment, is largely to be established. Thank you for your attention. You can always contact me at joris.veltman@ncl.ac.uk.